Now we are beginning chapter 8. So we basically set the evolutionary and ecological stage by going through the physics and the ways that habitats are distributed, the ways that different habitats can be formed. And now we're going to, in this evolutionary play, if you think about that way, we're going to introduce the players. And the players are the organisms. And the reason I do this at this point is that we need, even then we'll go back and talk about nutrients, but we need to talk about how organisms do things. So I need to be able to refer to organisms and make sure you're up to speed on the aquatic organisms. This first dendrogram has the organisms in the major groups, the eukarya, the bacteria, and the archaea. And we can see a number of things here. First, there's 3 domains of life. So when I was a student, it was just the eukarya and the prokaryotes, what they call them. But then they discovered this other group, first in extreme habitats, and then they found them, they were pretty much everywhere. These are probably, now they're thinking the archaea are probably the progenitors of the eukarya and ingested and somehow enveloped some organelles and became the eukarya with nucleus and organelles. Another thing we can take away from this immediately is that there's some things that occur in the eukarya, and we'll talk about this a little bit more, that clearly came from the bacteria. So chloroplasts, if you sequence the genetic material in chloroplasts, the organelles in plants, right, that allow for photosynthesis and green algae, they actually fall out down here with the cyanobacteria. They are bacterial. Likewise, mitochondria come out pretty close to Escherichia coli, one of our gut microbes. And again, they're found in the eukarya, but they actually originated from bacteria. Another thing that happens is when we talk about the major groups of eukarya, we talk about animals, plants, fungi, and then the protists. And the protists are everything down here. And there's a bunch more stuff there, but the other thing that becomes immediately obvious is that, you know, we are almost fungi, basically. We are so closely related relative to some of the protists that are very distantly related from each other, right? So we're just all a bunch of mushrooms, I guess, if you want to think about it that way, with legs. So as we go through the types of aquatic organisms, we'll talk about the species concept, the major taxonomic groups, how we classify organisms by functional significance instead of taxonomy or phylogeny, and organisms that we find in freshwater systems. So this sets the stage, and then we'll talk about microbes and plants in the next chapter, and then after that we'll talk about animals in the chapter following that. So here's another question for you. How do you define species? Somebody throw in a definition of species real quickly. Either verbally or type it in. Okay, good. Cassie got it. Can reproduce and are genetically similar. They can basically reproduce, and so that's the biological species concept you're talking about. How do taxonomists usually define species? And I think somebody actually got kind of got the answer already. Emma said a group of similar organisms. That's right, things that look similar to each other, right? And that's how functionally we define species. If we see 2 birds and we want to know if they're of different species, we don't test whether or not they can interbreed with each other. We go to a manual and look up what their feather color, their shape, their size, those kinds of things, and use those to define the species. The taxonomists obviously get more into that because they try to find characteristics that are actually related to phylogeny as well, and try to organize things that way. what is a major problem with requiring that two organisms that we're trying to decide if they're distinct species be able to reproduce? So why is it that biological species concept that if they can reproduce with each other, they're the same species, but if they can't, they're different species? Yeah, mules are an interesting one, right? So a horse and a donkey can reproduce, but their offspring can't, right? So that's you can think also about a dog and a wolf, right? We view those as separate species from each other, right? But they can reproduce with each other fine. They may not mostly, but they can. What about the vast majority of microbes? What's the problem with that? Particularly things like bacteria.
They don't sexually reproduce.
Exactly, no sexual reproduction, right? So there's many species that have no sexual reproduction. So how can you require sexual reproduction to be a prerequisite for species? If that's the case, then every single cell is its own species because it can't reproduce with anybody else, right? So there's another problem with that, right? So we have different ways that we can define species, and we need to think about it more generally. One of the things that's really helped us in that is the molecular methods that have come along. There are some problems with using the way an organism looks to tell whether it's a distinct species or not. So phenotypic variation can be a function of abiotic habitat conditions. In this example, this macrophyte here that's growing growing in a clear lake, when its leaves are underwater and develop underwater, they get these nice slender look. But when the plant gets tall enough to be emergent out of the water, it gets these wider leaves. You can imagine why do you think that it might be adaptive to have these narrow leaves when you're underwater? Streamlined, good, Lane. Yeah, sure. I mean, like if there's water currents in there, water's gonna be able to flow around. If you have these big paddle-shaped leaves, it might be a problem. But this plant might spread out and be more photosynthetic if it can do it without getting wiped out by the currents. So we'll talk about the major taxonomic groups, the archaea, the bacteria, and the eukarya. And then we'll talk about using ribosomal RNA for taxonomic identification of organisms. And we will do that in the next lecture because we have about 4 minutes left or 3 minutes left.